Thanks very much, Emma, and thanks to everyone involved in Step Into Oxmed. It's a fantastic initiative and we're very proud to support it and pleased to support it. And thanks to all of you for coming along this evening. I'm sure you have other things, other demands on your time. So what I'm going to do in the next 30 minutes or so is just tell you a little bit about my career to date in basic cancer research and highlight some contributions, important contributions that have been made by other people, which have allowed me to play a small part in the story that I'm about to tell you. And the title is a bit of a giveaway, really, because we're going to start in basic biology, in yeast and frogs, and we're going to hope that those experiments in those model systems will tell us something informative and useful about the biology of a very complex set of human diseases, which are the cancers. So in essence, what we're trying to do, I guess everyone can see the arrow. Is that right? Can you see the arrow?
I can't see the arrow.
Yeah, okay, right. I'll try and choose some other thing down here. Okay. I don't seem to have any options here that will allow me to give to generate a cursor. But anyway, what you can see in this slide, which was made by a clinical research fellow in my lab some years ago, Zoe Winters, is a thin section through a human tumor. This is actually a breast cancer. And Zoe's prepared this slide in a way that you can see the cancer cells quite clearly because she's stained them with an antibody that stains them brown. So the brown cells here are the cancer cells. And what is made very graphically obvious in this image is the fact that the cancer cells are invading the normal tissue. You can just see some normal tissue down to the lower left of this field here, and they're destroying that tissue. And it's this capacity of cancer cells to grow and multiply and ultimately to invade and destroy normal tissue that is the basis of this set of diseases. And it's what makes them such a terrible set of diseases. And we're interested specifically in the property of cancer cells that they continue to grow and divide and make more cancer cells, rather than the process of invasion and spread to distant sites, because it's that process of cell division that is really fundamental to this set of diseases. And just a quick refresher course to remind you of something that I think is probably already familiar to you. You know that we're made of cells. We're made of an awful lot of cells, around about 10 to the 14 nucleated cells in a human being. And they're all generated, of course, ultimately from a single cell, the fertilized egg. And so this is a vast number of cells. And many of those cells have jobs that don't involve them making more cells, but in particular epithelial tissues and bone marrow and various other tissues that are characterized by a continuous turnover of cells during our lifetime, the progression of cells through what we call the cell cycle. And it's the cell cycle that's illustrated in this slide here. And if we just start at the top, we have the process of M phase, which is in body cells mitosis, which gives rise to two daughter nuclei, which are separated into two daughter cells. And then we have the rest of the cell cycle, which consists of the synthesis phase, the S for synthesis phase down at the bottom, where the DNA in each chromosome is copied to generate a new chromosome. And usually gaps in between M phase and S phase, which are gap 1 and gap 2. And so there's this repeating cycle of events which generates more cells that is really the focus of our interest in research terms and has been for many years. So just as an aside, it's important to distinguish between this process of cell division, which generates more cells, from the process of cell growth, which makes cells bigger. And although many cell populations that are going through the cell cycle also get bigger as they go through the cell cycle, those two are quite separate processes, and that's sometimes an important distinction. So how can we learn more about this process of the cell cycle, which generates more cells, and generates more cells, of course, in perfectly normal tissues as well as in cancer cells? This is one way of visualizing the cell cycle process. These are just static images. I'll show you a movie in a second. And these static images generated by Uli Gruneberg in the Dunn School of Pathology in Oxford show live cancer cells. These are human cancer cells which have been engineered in the lab. So they express a green fluorescent version of the tubulin protein, which makes the microtubules in the cell. And they're also expressing a red fluorescent version of the histone protein that is part of the protein component of chromosomes. So although these cells are alive, if we illuminate them with the right wavelengths of light, we can see in living cells the microtubules, and we can see the histone proteins, and so we can see the chromosomes. And so what you see here is a series of images from left to right, going from what we call interphase, the part of the cell cycle that is not mitosis, and then in the second image, the cell is entering prophase of mitosis, and you can see individualized chromosomes in red starting to appear. And then in metaphase of mitosis, those condensed individualized chromosomes are aligning at the metaphase plate in the middle of the cell. And what you can see quite dramatically in that third image is the appearance of the mitotic spindle, which is made of tubulin protein and is bright green in this engineered cell. And it's that microtubular spindle which is responsible for pulling apart the segregated chromosomes during anaphase of mitosis, which is the fourth image here. And in telophase of mitosis, the two daughter nuclei start to reform. So that's quite a nice set of images, but it's much more dramatic when you see a field of human cells doing this in not quite real time. It's slightly speeded up, but in the video you're about to see, what you see is a small number of human cancer cells going through this process of the cell cycle. So I hope everyone can see that, and it'll probably go through a couple of times, but what you can see is cells in interphase, this cell on the left here, in interphase, which is going into mitosis, and it's only in the M phase of the cell cycle when these individualized chromosomes become apparent. And you can see the incredible reorganization of the microtubules, the cytoskeleton within the cell, to form this mitotic spindle. I think if we let it go around again, if we can, you'll see that happening again. It's worth watching twice, I think. Here we go. So just watch these two cells in the middle. There's one on the right going into mitosis. There's metaphase to anaphase transition. And you see how rapid it is. There is a time base up in the left-hand corner here. And this film has been generated by taking images every second or so of this field of cells, and then stringing those together into a movie. So it's obviously a very elegant and beautiful process at this level, although the fact that these are human cancer cells makes it slightly more sinister. But it's also very complicated, because we know there are many thousands of genes in each of these human cells, and many thousands of different proteins encoded by those genes. So how can we ever have a hope of understanding this complex process by which the cell cycle generates more cells? Well, surprisingly, most of what we know about this process in human cells comes from studies of what we call model organisms, much simpler organisms, which, although they're simpler, are still eukaryotic. In other words, they have a nucleus, and they share enough biology with us that we can learn a lot about human cells by studying them. So let's take a look at the first of those models. And this is a fission yeast, shown here on the left-hand side. These are a series of micrographs of these yeast cells. So it's a single-celled eukaryotic organism. And it just grows as these single cells. They look a bit like large bacteria, actually. They grow as this sort of cylindrical cell, which, when it reaches about 14 micrometers in length, will divide by fission, in other words, by chopping itself into two daughter cells. And that's the sequence that you see on the left-hand side there. And it's a very simple eukaryote. It doesn't have all of the features of multicellular animals or plants. But it does share this basic biology of the cell cycle, and it's that attracted the character on the right, who's Paul Nurse, to investigate the cell cycle in this organism. He reasoned that it would be genetically simple enough that we might be able to learn something about the cell cycle by studying this organism. It has a tiny genome by comparison with the human genome, which is about 3,000 million base pairs. This yeast has a 12 million base pair genome, so almost 300 times smaller than the human genome. And it's also not subject to sort of ethical concerns. And you can do all sorts of interesting experiments with the yeast, like induce random mutations into the genome and find interesting phenotypes, which means that the yeast is a very good model for what's called classical genetics, where we start out with an interesting phenotype, in other words, some interesting abnormal behavior of the yeast cells in this case, and from that interesting phenotype, we're able to identify eventually the genes that are responsible for controlling that process in the normal cell. I'll explain what I mean by that in a moment. So Paul Nurse has had a long and distinguished career, as you can see on the right-hand side, and it was during his Oxford years from 1987 to 1993 that I worked in his group, and it was a very productive and exciting time during which we learned quite a lot about how the cell cycle is controlled. And I'll tell you a little bit about that now. So the early work by Nurse had already identified some of the important genes that control progression through the cell cycle. So he identified these genes through classical genetics by identifying mutant yeast cells, which had the phenotype shown in the upper left-hand part of this slide here. And this is called the cell division cycle phenotype. What you see on the left-hand side, labeled WT, that's wild type. Those are normal fission yeast cells just growing and dividing. And the nuclei are stained here, so you can see the two nuclei in the cell at the bottom. And the cells on the right-hand side have a problem. They've been mutagenized. They've had random mutations introduced into their genomes. And by chance, this particular clone of cells has picked up a mutation which has an interesting phenotype. The phenotype is that the cells become blocked in the cell cycle. They continue to grow, so they get longer and longer, but they just have a single nucleus, as you can see that on the right-hand side in panel B. So the issue with these cells is that they've, by chance, picked up a random mutation in a gene which is important for the cell cycle. So if that were a permanent problem, then these cells would simply be dead. They wouldn't be able to go through the cell cycle and make any more daughter cells. But the mutation is even more specific in that Nurse selected for temperature sensitivity. In other words, a phenotype that could be switched on and off by shifting the temperature. So at the high temperature, you see the phenotype on the right, but at a lower temperature, the cells are able to grow and divide quite normally, which means you can keep them alive. And by identifying cell division cycle mutants of this sort, Nurse was able to identify genes which were required, for example, CDC13 here. Again, I'm not sure if you can see my cursor, but you can see CDC13 is labeled on this yeast cell cycle just before mitosis. That's because if you have a temperature-sensitive mutation in the CDC13 gene, the cells become arrested just before mitosis. And of those various CDC genes that are defined by these CDC mutations, you can see one of them is quite interesting. That's called CDC2 for cell division cycle 2. And that's required twice in each cell cycle. It's required once here at the beginning of mitosis, and again a second time in the gap 1 phase, where the cells decide whether or not to complete a new cell cycle. So that makes CDC2 look interesting. And there are other reasons for being interested in CDC2 that I don't have time to go into, but it looked like CDC2 encoded an interesting gene. But for the time being, Nurse had just a collection of yeast mutants. So how do you go from yeast mutants to identifying the genes that are defective in those mutants? Well, these days we just sequence the entire genome, but that simply wasn't possible in the 1970s when this work began. And so it was necessary to do something a little bit more clever, and that's called functional complementation. And the idea here is that we start out with a collection of all possible yeast genes just by smashing up the yeast genome into fragments, sort of bite-sized chunks, and introducing each of those chunks into a circular DNA molecule, a plasmid, and generating a large collection of different plasmids, each of which has a random fragment of the yeast genome from wild-type functional yeast cells. So the idea is that somewhere in this collection, this library of DNA fragments, there is a fragment that by chance corresponds to the functional version of the gene that might be defective in, for example, the CDC2 mutant that we just looked at. So if we can introduce that into our temperature-sensitive CDC mutant, then that gene, which I've color-coded green here, will be able to provide the function that is lacking in the CDC mutant when it's shifted to the high temperature. So first of all, we introduce the gene collection, the library, and then we identify the cell that by chance has picked up this complementing green fragment, the wild-type copy of the gene, by shifting the temperature to 36 degrees. And if we do that, because these yeast cells are temperature sensitive, they'll arrest in the cell cycle unless they've picked up a fragment of DNA that allows them to complete the cell cycle. And only the green colony in this case, this is supposed to be an agar plate covered with yeast colonies, where each colony represents the clonal progeny of a single cell which has picked up a single fragment of DNA from this library. So the colony that can grow at 36 degrees should be the one that contains the gene that we're looking for. And so we just smash that colony open and identify the gene that is encoded on that plasmid molecule. So it's a bit esoteric, but if you follow the logic, maybe by looking at the recording, you'll see that this is really quite powerful, because the yeast are doing all the work. They're showing us really which is the gene that allows them to grow at 36 degrees, and the inference is that's the gene that is defective in the original mutant. And the great strength of this approach was that it's possible to use any source of DNA. And one of my colleagues, Melanie Lee, in the 1980s was able to, instead of using yeast DNA for this experiment, using human DNA, was able to perform this experiment where human DNA corresponding to human genes was introduced into the same CDC2 mutant. And through that process, it was possible to identify a human gene that was capable of providing the function that was missing in a CDC2 mutant. So this is very strong suggestion that whatever the CDC2 gene is, it's very highly conserved in evolution, right away from yeast all the way through to human cells. And the reference is down here in the lower left, in case you want to look at the original paper. So this suggests that CDC2 controls the cell cycle at 2 points in the yeast model, and is a very highly conserved gene that's present in human cells. So to cut a long story short, it turns out that this gene, which was originally called CDC2, is really remarkably highly conserved across evolution. So what I've done here is to just align the protein sequences, the amino acid sequences of the proteins that are encoded by two different yeast CDC2 genes, so from S. pombi and S. cerevisiae, and also the human gene that was identified by Melanie. And so each letter here represents an amino acid. So E represents glutamic acid, for example, and G is glycine. And where the amino acids are shaded in, they're identical in all three of these proteins, even though we shared a last common ancestor about 1.5 billion years ago. And there aren't many human proteins that are conserved, or proteins in eukaryotic cells that are conserved to this degree. And also that the experiments that Melanie had performed suggested that the function as well as the sequence of this protein was very highly conserved in human cells. So my contribution at this point was to identify amino acids in this protein that were regulated by reversible phosphorylation. There's a threonine and a tyrosine here and a threonine here, which allow the protein to be turned on and off. And the function of the protein is to act as an enzyme. And it's an enzyme that works by taking phosphate from ATP. I'm sure you know quite a lot about ATP already, the universal energy molecule of the cell. And this enzyme takes phosphate from ATP and attaches it covalently to its substrate targets. And in doing so, it modifies the function of those proteins. And so we'll return to that idea in a second. But before I get there, I just want to introduce the second model, explaining the reference to frogs in the title. And this is a completely independent strand of work, but it's again begun in the 1970s. And it's initiated by Yoshio Misui, who worked most of his career in Canada, shown in the upper left here. And Misui was interested in what at first looked like a rather different problem or question. He was interested in the process of egg maturation in the frog. And that's normally something that is triggered by progesterone, which you can see here in the diagram in the cartoon. And progesterone stimulation in the natural environment of the frog will stimulate oocytes, immature eggs, to re-enter meiosis. This is the specialized cell division that gives rise to germ cells. And they'll then become arrested in meiosis 2, metaphase, waiting for fertilization. As soon as they're fertilized, then that the zygote will go through rapid cleavage divisions in the early stages of embryogenesis. And Masui's observation was that you could extract oocytes from the frogs surgically, and then you could bypass the requirement for progesterone stimulation by taking cytoplasm from the mature egg and injecting it into the immature oocyte, and that would induce this process of meiotic maturation. And that's possible because these cells are huge by comparison with our own cells. These cells are about 1 millimeter across. If you ever looked at frog spawn, you know, there's little black dots in the frog spawn are individual cells. Each of those is a mature egg cell, and that's the sort of scale which allows you to, first of all, use a small needle to extract cytoplasm and then to inject it into oocytes and ask whether or not that can induce maturation. So using this very fiddly assay, Masui was able to plot the appearance of what he called maturation promoting factor, this mysterious substance in the mature eggs, which was able to bypass the requirement for progesterone. And he noticed that that's plotted in cartoon form here by the red line at the bottom. And he noticed that this maturation promoting factor, which he was describing in this rather vague term at this point, appeared every time the cells went through M phase. And so it appears during meiosis 1, then disappears again as the cells leave meiosis 1. And as they enter meiosis 2, it reappears. And on fertilization, it disappears very rapidly and then reappears again every time these early cleaving embryos go through M phase. So although he had no clue at the time about what this stuff was, he reasoned that it had the properties of an enzyme that was driving M phase entry, either into meiosis or mitosis. And another scientist, Jim Maller in Colorado, was able to build on that work by taking large numbers of frog eggs. Here's the African clawed frog, Xenophus laevis, which is where all these frog eggs come from. And he just centrifuges those eggs at low speed, and they're huge, because they're such huge cells, they they sort of collapse under their own weight and generate this pure undiluted cytoplasm. So there's no dilution of the biological material here, and this pure egg cytoplasm turns out to be a very rich source of all the things you need to go through the cell cycle. And Maller realized that it should be possible, in principle, to use this material to identify the maturation promoting factor. And he was able to show that we could use this pure cytoplasm to reproduce many of the events of the cell cycle, even though the cytoplasm is not cells. This egg extract is a so-called cell-free extract. It's possible for that extract to build a nucleus. If you give it some DNA, it will build a nucleus around it. It will replicate that DNA once, but only once, and then it will go into a sort of mitosis, even though it's not a living cell. And that assay allowed Maller and co-workers with Fred Locher to purify this maturation promoting factor because it was able to induce mitotic events in this egg extract. And that's pretty much where I came into the picture. I was able to take Fred and Jim's purified MPF and show biochemically that it contained the frog equivalent of the CDC2 protein that had been found previously through the yeast genetic approach that we outlined. So it turns out that both of these very different lines of investigation, one genetic in the yeast, one biochemical in the frog, give rise to precisely the same answer, which is that the enzyme that drives entry into M phase is this protein kinase enzyme. So it's adding phosphate from ATP to substrate proteins, and it turns out the part of the protein that's encoded by the by the CDC2 gene is the protein kinase, shown here in blue on the left-hand side, binding to ATP, which is this magenta stuff. But it only works if it's binding to a second protein, shown in green here. This is a cyclin protein, which was identified initially by Tim Hunt working in surf clams. There's another model system which contributed importantly to the identification of this accessory subunit. And because the kinase only works in the presence of the cyclin, it's called a cyclin-dependent kinase. And this dimeric, this two-protein enzyme is otherwise known as cyclin-dependent kinase 1, CDK1. And it's this thing that drives all of the visible processes of mitosis that you saw in the movie earlier on, like condensation of the chromosomes and assembly of the mitotic spindle and so on. So this is the enzyme that catalyzes mitosis, in other words. And just to sort of fast forward a little bit, in the interest of time, we now know that the cell cycle, as I've introduced it previously, is actually driven by the sequential activation of different cyclin-dependent kinases at different times. So the CDK1 cyclin-dependent kinase that I just described is responsible for catalyzing entry into mitosis. And then there are two other CDK-dependent events. The first is when cells decide in the GAP1 phase that they're going to complete another cell cycle. So that's called cell cycle commitment. And then there's a second, or rather a third, CDK-dependent event, which is the initiation of DNA replication itself when cells start S phase. So each of these is dependent on these protein kinase enzymes that are dependent on the cyclin proteins. And in the yeasts, it's relatively simple in that there's a single CDK enzyme that is sequentially picking up different cyclin proteins as the cell goes through the cell cycle. And in human cells, it's a bit more complicated because there are multiple different cyclin-dependent protein kinases. So CDK1 is this conserved CDK that was identified by Melanie that drives entry into M phase, probably in all eukaryotes as far as we can tell. And then other CDKs are responsible for the process of cell cycle commitment in the GAP1 phase, which in mammalian cells is called the restriction point, and the initiation of DNA replication. It's this restriction point that is particularly relevant to the cancer problem, because we know with a high degree of certainty that cancer cells very often lose this restriction point control. So the reason they're generating more cells is because they're constantly committing to a new cell cycle, even when that's inappropriate. And that process of commitment to a new cell cycle is thought to be catalyzed by a cyclin-dependent kinase called CDK4 or CDK6 in collaboration with a cyclin protein called cyclin D. And just want to give you a taste of the sort of state of the art in terms of the technology that can be applied to biological problems or medical problems these days. And that revolves around sequencing the genomes of very large numbers of tumors or normal individuals. And it's through the application of this machine on the left-hand side here, an Illumina DNA sequencing machine that can generate these vast amounts of data shown on the right-hand side, which means that we can very quickly and easily describe all of the genetic changes that occur during the development of the tumor and identify the genes that are changed during tumor development and progression. And if you're interested in this sort of science, big data science, you can look at the reference by Campbell et al. Shown on the left-hand side here. You see it's quite recent. And what you're looking at here is just a league table, if you like, of the genes that are most frequently mutated, changed in human cancers. And they're relevant to a wide variety of human tumors, which are listed along the top here. So we've got liver cancer, prostate cancer, breast cancer, and so on. And the gene names are listed top to bottom. And several of these genes are involved in that restriction point control. So here's one called RB1, which is a negative regulator of restriction point control. It's very often mutated in different tumors. This one here, CCND1, encodes the cyclin D protein that I just mentioned, which is the cyclin protein that catalyzes commitment to the cell cycle. And CDKN2A and CDKN2B are inhibitor proteins that inhibit the cyclin D-dependent protein kinase. So these genes are frequently mutated because they allow cancer cells to commit in an unregulated way to new cell cycles. So I'm interested in particular in the cyclin D1 down here because that's the component of a cyclin-dependent protein kinase. And in recent years, small molecule drugs, so these are drugs that have been developed in part on the basis of knowledge of the three-dimensional structure of the cyclin-dependent kinase proteins. And these drugs are all approved in the US and the UK for breast cancer patients. So they're competitive inhibitors of the protein kinase. They look a little bit like ATP, and they compete with ATP for binding to the CDK enzymes. And they work. They have measurable clinical benefit. So the red line here, this is for ribociclib, one of those inhibitor drugs. You can see that this cohort of breast cancer patients, it does rather better in terms of overall survival than the standard of care, which is the blue line here. And here's the corresponding set of lines for one of the other inhibitors, abemaciclib. So this is a new class of drugs. Notice these publications are quite recent, which can be offered to breast cancer patients whose tumours are estrogen receptor positive. So this is a great advance. But just to finish, I just want to raise a bit of uncertainty, because although these drugs offer clinical benefit, I'm personally not convinced that we know quite how they work. So these are the drugs. How do they work? What might explain those clinical responses that I just showed you? And it's made complicated by the fact that we now know that the human genome encodes a huge number of different protein kinases. We know there are 518 of them. And in amino acid sequence, they're all quite similar to each other, and their ATP binding pockets are all very similar to each other. And so there's a very real chance that any drug that works by competing for ATP binding to a protein kinase is actually hitting multiple protein kinases all at once. And that's sort of important because recent evidence that we have suggests that although this is the sort of textbook model of how the cell cycle is controlled, we know that this cyclin-dependent kinase 1, or CDK1, can actually substitute for these other CDKs and might be itself very directly relevant to this process of commitment to the cell cycle. And so it might be that we should be, instead of targeting CDK4 and CDK6, which against which those inhibitor drugs were designed, we might do better to target CDK1 instead. So I'll leave you there with some conclusions. What I hope I've shown you is that simple experiments using yeast genetics can identify important conserved regulators of the cell cycle, such as CDC2. And this independent line of inquiry, biochemical approach in frogs, identified the mysterious biochemical factor, maturation promoting factor, which turns out to be just the frog equivalent of CDC2 plus the cyclin protein. And it's these CDK cyclin enzymes that drive the key cell cycle transitions in probably all eukaryotic cells. And they're dysregulated in particular ways in tumor cells. That means that inhibiting those enzymes can be therapeutically useful. And by understanding more about the basic science, we might be able to target those drugs more precisely to improve the magnitude of those clinical responses. So that's all I'm going to say this evening. This is where we work. This is the Oxford Molecular Pathology Institute, which is quite a flashy, relatively new building. I've been supported by Cancer Research UK through most of my career, and none of this happens in isolation. This is all work that has been done by large groups of individuals, and these are the people who have worked with me over the years on the projects that I've described. So I'm going to stop there and maybe unshare, and I'm going to ask Emma to field any questions that you might have.
Great, thank you, Chris, for that talk. It was really interesting. I've always found it quite interesting to see how learning from really simple organisms can be scaled all the way up to the point where we can make medicines for humans. It's really interesting. So now you guys can ask any questions you have on the talk, or maybe about Chris's career, anything you want. There's a Q&A tab that you can send questions into. There's also the chat that you can use as well. So I guess we'll give it a few minutes, and whilst we wait for people to ask questions, maybe I'll start. Chris, how did you get interested in molecular biology as a career? What inspired you?
That's a great question. So I have to admit, I was an undergraduate in biochemistry, and I was not a very conscientious undergraduate at first, because I found it all a bit confusing, and some of it was a bit boring as well, to be honest. But then I had the opportunity as part of the course to do a research project, and I was absolutely in love with it almost from day one. And that was in 1982, which is still the very, very early early days of molecular biology. And so I got to sequence a bit of DNA as part of that project. And I just thought that was the most exciting thing possible. And it's sort of trivially easy to do that now, but it was very difficult in 1982. And I got to do some simple experiments that told us something we thought about DNA replication. So I was already working on the cell cycle in some respects by then. And I just was totally hooked within a couple of weeks on just the idea of generating new information about something that nobody had come across before. You know, the DNA I sequenced had never been sequenced by anybody before, and I thought that's quite a rare opportunity. You know, to see something that nobody's ever seen before. You know, you could travel the whole world and you struggle to find somewhere that nobody has seen before, right? And most of us don't get to fly in space and so on. So this is the next best thing. Maybe you get to discover something that nobody has ever known before. And so I just asked my tutor at the time where was the best place to go in the UK to learn more molecular biology, and he suggested what at the time was called the Imperial Cancer Research Fund, which is now Cancer Research UK. So I went there in London. For 4 years, I worked there. I worked on a tumor virus initially, so a virus that causes cancer in mice. And Paul Nurse was working on the same floor in the same institute, and so I got to know members of his group at that point, and took it from there, really.
Lovely, thank you. We've got 2 questions. The first question is, why don't frogs have CDK1?
Well, they okay. Thanks very much for the question. So your question probably reflects the fact that I spoke too quickly. So frogs do have CDK1. So CDK1 is the same thing as CDC2. In the old literature, it was originally called CDC2, and it's because it was called various different things in different organisms. It was decided to give it a sort of standardized name, which is CDK1. So the enzyme that was purified as maturation promoting factor from the frogs is CDK1, and the enzyme that is encoded by the CDC2 gene in the yeast model is also CDK1, and it's the same enzyme that was identified by Melanie through this complementation cloning strategy from human DNA. So it's all the same thing. It's just had different names at different points during the development of the field.
Great, thank you. And the second question is, what sort of grades would you need to achieve in order to get into this career?
Well, I think there are various routes into this career. I think all of them are fairly competitive. You can take a route through basic science, through undergraduate courses like biomedical sciences or biochemistry, and those are already quite competitive courses. I think the standard offer for biomedical sciences here is A-star AA at A level. So those sorts of grades are going to work for you. I mean, not all biomedical sciences or biochemistry courses are quite so competitive, but you're probably going to need, I would say, at least 3 Bs at A level to compete successfully for one of these courses. Or you can take the medical route. You can train in medicine, and that has some advantages in that the career structure in medicine is much more precisely defined. So once you've qualified in medicine in the UK, although you'll be aware of all of the discussion around medical careers and pay, the pay scales and so on for medics, it's still a very well-structured career in comparison to research science, which is much more sort of random, haphazard. So training in medicine first is quite a clever way to leave open the possibility of a career in research. And quite a lot of people who graduate in medicine are able to find a career sort of portfolio which includes some element of research. And depending on what type of research you're getting into, having a medical background can be a huge advantage because you have this greater level of understanding. Say, and they're all a little bit competitive. Some are slightly more competitive than others, I suppose. I hope that helps.
Yeah. One thing I didn't realise when I was in sixth form is that there are so many different ways to get into lots of different careers. So I guess keeping an open mind and researching a lot and asking people can help you kind of figure out, you know, the ways you can do things. In the chat, I'm going to put a feedback form for people that want to fill it in. Let us know what you liked, what you didn't like, and what kind of talks you'd like to see next. In the meantime, we do have another question for you, Chris. That's okay. This question says it's slightly unrelated, but what is the role of the P10 protein, PTEN protein?
That's a good question. And so P10 is a so-called tumor suppressor. So it's a protein that, when it's functional, is good for us because it tends to inhibit the development of tumors. But when it's mutated, when it goes wrong, that generates a cell that is more likely to become a tumor. It's a really interesting protein that's encoded by the P10 gene. It's a phosphatase protein. That is to say, it does precisely the opposite of protein kinases. It actually removes phosphate from proteins rather than adding phosphate to proteins. But it also removes phosphate from lipid substrates. So it's a bifunctional phosphatase, and its most important function in cancer terms is probably to reverse the lipid phosphorylation that is carried out by another enzyme called PI3 kinase, which is a lipid kinase. So the more you get to know about human cell biology and cancer cell biology, the more apparent it becomes that large amounts of our biology are governed by reversible addition of phosphate to substrates. So addition of phosphate to lipids in this case, but also reversible addition of phosphate to proteins. So it's almost universal. It's used for all sorts of different signaling processes in cells. And many of them are relevant to cancer. So yeah, that's a long way of saying that P10 is a lipid phosphatase.
Thank you. We've got two more questions. The first one is, are there any more sessions? Yes, there will be. We plan to hopefully do maybe one a month, and we're open to suggestions from you guys from what you'd like to hear about. So make sure you fill in the feedback form so that we know. But next month we'll be having a respiratory doctor. So that's a doctor that looks at the lungs to talk to us for a little bit. So I'll email that all out to you so you can join if you'd like. The second question is, what grades would you say you need to take medicine in university to become a surgeon? So I'll just take that quickly. Most universities, it'll be 3As or A-star AA and maybe more. That like 3As, A-star AA is the standard for medicine, but other universities do have kind of contextual offers or offers that can be altered depending on maybe your EPQ. So definitely do some research online to see which universities are best suited for you. There's lots of free online resources you can use like MedicMind and things like that. That would be good. It doesn't seem like we have any more medical questions. Someone asked, will we be sent the recording of the session? And I'll put it up on YouTube as soon as I can so you can rewatch it if you'd like.
Okay, well, if there are no further questions, I just want to say thanks very much to everyone for turning up. And thanks very much for your questions. And I hope some of it made sense. And I hope you're taking something interesting away from this. I guess if the recording is available, that will help you to revise it at your leisure. Anyway, very nice to meet you all virtually.
Thank you, Chris, and thank you all for coming. Bye.
Bye now.